thank you so much for the invitation to be here. I'm really excited to chat with you a little bit. I sort of threw in a little bit of everything. My path to the kind of research that I do today, a little bit about some of the research that I have done in the past, a little bit about the research I do today, and how I connect that in the classroom for my students at University of Richmond, and some teaching initiatives that I've been a part of. So in high school, I knew I liked being outside. I grew up in the woods, and when I got to Davidson College in North Carolina, I connected with some biology professors that were doing research outside. And, you know, I thought, hey, I'd like to go to Africa and study gazelles, or I'd like to go to Australia. But I learned very quickly that there's a lot of amazing flora and fauna in my own backyard. And so while I was there, I actually got involved in the lab that studied amphibians and reptiles. And if you told me, like, when I was in high school that I would be holding a rattlesnake on the end of a hook, I would not have believed you in the slightest. But I found a passion in particularly turtles when I was an undergraduate. They're easy to catch, they're really fun to work with. You can find them in ponds all over the landscape. There are a lot of interesting conservation issues with turtles, and so I did a little project where we attached these these temperature data loggers to the backs of the turtles and looked at their activity based on their temperatures. And I discovered that you could actually make a career out of doing research, which was stunning to me that you could actually make a career out of doing this kind of work. So I was so passionate about it and so excited, I went straight to graduate school. I ended up back in Virginia at UVA. Has anyone seen the movie Dirty Dancing?
Yes.
Yeah. Are you aware that Dirty Dancing was filmed in Southwest Virginia? Yeah. Mountain Lake. So Mountain Lake Biological Station is literally just down the road from Mountain Lake Hotel, just outside of Blacksburg in Southwest Virginia. And it's like summer camp for scientists. It's this glorious place. They have this big stone building made out of stained stone. There's the fancy hotel down the road, a big complex of cottages, classes, scientists who spend their whole summer doing research there. And I spent 5 years there studying red-spotted newts, which make migratory decisions between different ponds. So some years they stay in the pond to breed, and some years they wander back and forth between the forests. So I spent 5 years following about 3,000 newts that bred in these complex of ponds and doing some experimental work looking to see what impacted whether they migrated or not. So after that, I graduated in 6 years, more school. And then right after I graduated, my daughter Grace was born. She's turning 7 on Sunday, which I did not imagine. But I did want to highlight that my career path also took some time to be a mom, which I was really grateful for that opportunity as well. So after she was about a year old, my husband and I, who's also an academic, he's a chemist, we decided we were up for a bit of adventure. So I thought about the kinds of research questions that I was interested in, and everything that I'd done up to that point was sort of population, particularly at the pond scale. And so I was interested in, going a little bit bigger, and so I found a research system in New Zealand on this amazing creature, the Tuatara, and wrote a grant, and we got really fortunate and packed our bags and went off to New Zealand for a year. And we lived in Wellington, on the very tip of the south of the North Island. And I thought I'd tell you a little bit about New Zealand and the project that I did there. New Zealand is a fascinating place for biodiversity. One reason is because it's had no snakes or terrestrial mammals in the whole of its evolutionary history before we got involved. So that means there is an incredible diversity of species that you find nowhere else. Over 100 different species of birds, until about 1,000 years ago when the Polynesians arrived. And they brought with them the Kior, or Pacific rat, which invaded much of the land mass. They also started eating and hunting a lot of the big birds that were found in New Zealand. And then in 1769, Captain Cook arrived, along with other Europeans soon after. And over 50 different mammal species were introduced into New Zealand after this time. And some really strange things like moose You can throw them on a big boat. So there are still about 30 different species of mammals found in New Zealand, both domestic, like sheep and cats, but also introduced, like brush-tailed possums from Australia, stoats, a lot, weasel kinds of creatures, a lot of things that like to eat the native fauna. So in this time, 30 different species of birds have gone extinct, including all large species like the moa, which was this huge, flightless, ostrich-looking bird, which was like 6 feet tall. It's had a huge impact on things like kiwi, like really charismatic species. Also insects, so if you've seen the Lord of the Rings movies, the studio that does all the special effects is called Weta Works. So it's named after this great insect that's found in New Zealand, like this giant cricket. Those are actually my hands. It was really cool to see them. And they're extinct on the main island, so you can only find them on offshore islands now. The creature I cited, the Tuatara, is also extinct on the main islands, and 3 of 7 species of frogs are extinct. And of the species that are remaining, 2 of those are only found on a single island. So it's kind of a repeat story, no matter what kind of organism you look at on New Zealand. A lot of them are only found on restricted islands, often only in very small numbers. So this was the reptile I studied, the Tuatara. Has anyone ever heard of the Tuatara before? What? Good. Its lineage is over 200 million years old. It's sometimes called a living fossil. That doesn't mean that it hasn't changed over evolutionary time since then, but it's a remnant of what this lineage used to look like. And it survived only in New Zealand for 80 million years. So just to give you an idea where Tuatara fall in the sort of reptile tree of life, if you think about the general classification of reptiles, reptile is a class in the kingdom animals, in the chordates, the animals with backbones. Does anyone know some major orders of reptiles? Throw out some ideas. If you were going to group reptiles into different classifications. Anyone want to throw one out?
Like the snakes?
Snakes. Okay, snakes is one. Another big group.
Lizards?
Lizards. Turtles. Crocodiles. Yep. So it turns out there are 4 main orders of reptiles. Crocodiles is one. We have about 23 different crocodile species on the planet today. Turtles is another group. They form this branch of the reptile evolutionary tree. And actually, you may know that dinosaurs and birds are actually descended on the evolutionary tree from reptiles. So when we talk about evolutionary relationships, we often talk about birds being a paraphyletic group because they're not actually their own group separate from reptiles. Then on the other branch of the reptile evolutionary tree, we have lizards and snakes forming one order, the squamates. And then we have a single representative of the order Spinodontids, which is the Tuatara. So it is the only living member, only living species on our planet that represents 220 million years of evolutionary history. So that's pretty cool. There are morphological characteristics that it has that are found in no other animals on our planet today. So it's certainly an organism that we're very interested in conserving. It's also a very iconic species in New Zealand. They're very proud of Tuatara. It has cultural significance to the Maori culture there. It's only found on these red and yellow dots here. The black dots are records from the fossil record, and these go all the way down to the bottom island of New Zealand. So we know that historically Tuatara were everywhere in New Zealand, but today they're only found naturally on these red dots, and then these yellow dots are places where people have created sanctuaries and fenced out all predators. So New Zealand actually has announced this huge initiative that by 2050 they want to try to rid their entire country of invasive mammals. It's a huge effort. It's never been done on this scale before. Normally people would put up a fence on this little peninsula and try to get the little peninsula free of introduced predators, or get one of these little islands that rats have invaded free from predators. But they think they're going to try to do it all up and down the country. So stay tuned if they can come up with the resources and the way to tackle that problem. So there are these reintroductions to try to reintroduce Tuatara across their range. These guys are incredible. They share a lot of characteristics that you might think of with turtles. They're long-lived. They can be over 100 years old. They mature really slowly. It takes 10 or 20 years before they are ready to breed. They breed infrequently, every 2 to 5 years. So these kinds of characteristics are typical of organisms that are particularly susceptible to slow recovery when their populations get small. They're nocturnal, they're terrestrial, and they're actually cold adapted. Something I never realized is how close New Zealand is to Antarctica. It is really cold there. And these guys kind of look like iguanas if you think about them in tropical climates. No, these guys really like to hang out in the cold. They only come out when it's relatively warm by New Zealand standards, but I still had to have a long coat on every time I was chasing them around. Another really cool thing about Tuatara is they have temperature-dependent sex determination, like a lot of reptiles. So we typically think about sex determination being chromosomes, right? You have an X and a Y, and whether you have 2 X's or 1 X and 1 Y determines whether you are a male or a female anatomically. This is not the case for a lot of reptiles and some fish and some other organisms. They have no sex chromosomes. If you took a DNA sample from a Tuatara and mapped out the chromosomes, you would not be able to tell whether it was a male or female. Instead, the temperature of the nest determines whether they're going to be a male or a female, whether they develop male reproductive organs or female reproductive organs, just like crocodiles. So it's really common in crocodiles, and it occurs in all crocodile species. A lot of turtles, it's less frequent in lizards and fish. Right now, it's not been found in birds, snakes, mammals, or amphibians, but you never say never in science. In like 2 years, someone's going to publish a paper in Nature and tell us that they've found it. And there are all these different patterns of temperature-dependent sex determination. In some species, you get females at lower temperatures, and you get males at higher temperatures. In other species, you get males at lower temperatures and females at high temperatures. And crocodiles have this pattern, where you get females at the extremes and males at intermediate temperatures. So Tuatara have this first pattern. They're one of the only species that actually have this pattern, where you get males at warmer temperatures. And so I'm guessing you can probably guess my punchline. If you get males at warmer temperatures, and we're living currently in a warming world. But don't wreck Romeo. Yeah. Okay, so like I said, there are a lot of Tuatara populations out there, over 30 natural populations and more being introduced every day. So the story I'm going to tell you about is just about one of those populations. We've done some surveys on other populations, and other populations are doing pretty good. This population is literally a chunk of rock in the middle of the ocean. It is tiny. It's about 4 hectares. There are between 300 and 500 Tuatara on this island. So they are all crammed in there. They are highly competitive. There's not a lot of resources. There's nowhere else to go. There's nowhere really to dig their nests and really good habitat. So it's a high-density population. And in the late 1990s, people started to notice that they were getting a lot of boys on this island. So we started doing more frequent surveys, or the group that I was working with started doing more frequent surveys. And I came in to help summarize some of their data and look and see if this male bias had gotten worse in the last 2 decades. And lo and behold, it has. So if we look at the surveys that were done over the past 20 years, and the number of males compared to females captured, so with a 1-to-1 sex ratio, you get captures on this black line, and in the 1990s, you can see you catch about 1.5 males for every female, and in the 2010-2012 range, we were getting over 2.5 males for every female we would catch. So that basically means the island's gone from about 60% males to about 70% males. And for a species that reproduces as slow as these guys do, that's a pretty big shift. So this raises some alarm bells. So we started thinking about what the trajectory of this population is and why the population is becoming more male biased. So one reason is I told you they're crammed in on that island. There's not a lot of food. they start competing for resources, and when we look at the body condition of the animals on this island, it has decreased over time, but it's not decreased at the same rate. So this is, again, year on the x-axis and body condition on the y-axis, and you can see that the decline has been steeper for females than it has been for males. Okay, so they're getting skinnier, and the females are getting even more skinnier. So then that means the females are laying less eggs. So in neighboring islands, they breed every 2 to 5 years. On this island, they breed every 9 years, which is incredible. Like, you go there, you want to survey and find some eggs. Only about 4 or 5 females are actually laying eggs in any given year. And instead of laying close to 9 eggs, they're only laying 6 eggs. So they're not laying very many eggs, so you're seeing decreased reproduction. And then, when they do lay those eggs, are the babies more boys than girls? So we can't really catch the baby Tuatara. They're really tiny. They disappear into the vegetation. It's really hard to catch them. So what the folks in this research group did is they used soil temperature data loggers to record what kinds of sex ratios you would expect to get given the temperatures of the soil. And they found under the current climate, you get about 56% males. So it's male bias, but not too bad. But then if we start seeing more warming, more increased temperatures under these different scenarios that the International Panel on Climate Change gives us, if we stick to this minimum warming scenario, you're not going to see a lot of change. But if we have the more 4 degrees Celsius increase under some of the maximum warming scenarios, that range, where that switch gets flipped to go from females to males, will flip towards all males. So that's of course a concern. But thankfully, because these guys are so slow to reproduce, we have some time before some of these dramatic scenarios take place. We also see some differences in survival. So being really long-lived, they survive a long time. Their survival is up around 98% every year. Very few are dying every year. But over time, this has decreased as that competition has increased, and you can start to see some differences between males and females, which might be contributing to the sex ratio bias. So this leads to what we're calling an extinction vortex. One factor sort of feeding on another to potentially create an all-male population by about 2150. We've got some time to figure out how to deal with this. But it is a sort of case study for what could happen for a species with temperature-dependent sex determination in these warming climates. So after I did this project and then started doing research back here in Virginia, I thought, how can I bring this to my students? Because I'd love to take them home to New Zealand, but I can't quite afford that. So I learned about an initiative to try to bring data into the classroom. Has anyone ever heard of data nuggets before? Have you used them in your classes? One person? So I met the authors, or the initiators of Data Nuggets from Michigan State University, who work for the Beacon Center for Study of Evolution. And the website is here, and of course we'll post this on the website afterwards as well. But it has a lot of great resources for bringing little digestible pieces of data into your classroom. So I thought I'd tell you a little bit about it, and then we could look at the Tuatara data nugget that I wrote with these guys. So this is part of a sort of bigger picture idea about thinking about teaching quantitative reasoning in science classes. So it's trying to get students to think about the world through mathematics, thinking about approaching everyday problems with logic, bringing in statistics to understand authentic situations, to understand when they see graphs on the news, when they think about their own health, and how to be a constructive, concerned, and reflective citizen who can incorporate quantitative reasoning into decision making. And this has been a shift that I'm sure you all are aware of in moving away from rote memorization of facts towards more critical thinking, to get students to analyze and interpret data, thinking about how they communicate arguments based on evidence, and bringing this mathematical thinking into science standards in the classroom, both at the middle school, high school, undergraduate level. In the classes I teach, we talk a lot about vision change in biology education. So with this idea that we want to be teaching this way, but how do we actually go about doing it? How do we provide resources for people to do this? In ecological data in particular, there's a lot of complexity to resolve. The data is really messy. If you went to the scientific literature and tried to pull a case study for your students, it gets really complicated. So, but these students need practice working with data, graphing and interpreting and thinking about it. So it's nice to have a more small, digestible chunk. So Data Nuggets was founded to bring free activities based on real data into the classroom that brings some of the messiness, but not too much of the messiness and complexity. They're based on real research projects, just like the Tuatara research project I told you about. And it starts from the background of the science all the way through to data analysis and interpretation. And they aim to be sort of 30 to 60-minute classroom activities, and they all sort of follow a similar template so that if you do one with your students, then you can get them used to the idea and bring in more and more as you find appropriate in your classroom. So this is what the website looks like. They have standards alignment and a lot of other educational resources on their website, as well as the data nuggets themselves. But here's an example of some kinds of data nuggets that you might look at when you go on the site. A lot of them are more ecological based right now, but I know they want to expand them. They provide different content levels, which I'll show you in a minute. And you can see the sort of keywords and summary of the research and the question that the students might think about. So the Data Nuggets format starts with background information about the research system. These students identify hypotheses and scientific questions associated with that research. They also have a great spotlight on science, and they try to attract scientists to work with them that are not your typical white-coated statesman scientists, that are more relatable, that represent the diversity in science. So you can actually see sort of the researchers and some of their stories as well. And then they also have an interactive map, so you can see where these projects are taking place. And you can Google Map and do a street view or a satellite view of the research sites. So there are data nuggets at every content level: elementary, middle, high, advanced, or AP high school students, up to college undergraduates, and they correspond with the science content standards. And each of them are also kind of adaptable, so you might take a data set and try to scale it up for your classroom or scale it down depending on where your students are at. They all have a data and data visualization component where the students are given the data and then asked to graph it or represent it or do some calculations like averages. And for more advanced classrooms, there are statistics extensions that provide students with doing the calculations for summarizing their data and looking for statistical differences between groups, for example. There's also 3 different sort of levels of what the student worksheet can look like. The data can already be displayed for them. The access labels can already be provided, the scale provided, so all the students have to do is interpret, all the way up to the students have to label the axes, identify where the data goes, and graph the data themselves. So it's really nice that sort of those levels are already scaffolded into the activity for teachers to use. There are teacher notes that provide background information. There are some checks for understanding where you can stop and check in with your students and see how they're doing. And there are also moments for the students to reflect on what they're working on. Also additional resources, so you can find the primary scientific paper. You can sometimes find videos that relate to the scientific system, popular press, and then of course the scientific paper itself. So some of the themes that are in Data Nuggets, thinking about science stories, so what the scientists wrestle with in the field themselves, what their goals were, the reasoning behind their experimental designs, active learning instead of just memorizing the facts. They're thinking about the process of science while you're doing the activity, And like I said, you can connect it with a particular place where that science was done. So there is one actually from Mountain Lake Biological Station on juncos, a small songbird. So that's a more local project that you could bring into your science class. Here's an example of some of the US-based data nuggets that are available. So this is the data that's in the data nugget that I wrote. I've brought you guys some copies of the student handout, the teacher handout, and how you might assess the student answers and the approach that Data Nuggets tries to get the students to think about the data. I wanted to highlight the interpret the data part, where we ask students to make a claim that answers a scientific question. And I sort of wanted to follow up on this thinking. The thinking is that it's important to ask students not only to identify what the scientific question is and the relationship between the variables and the data, but how scientists use that data as evidence, and how they then reason through a claim that they might make from that data, and how you connect the data with the hypothesis. So the page on, let's see, I think it's right after, here we go. I've sort of printed out an example of how you might have your students walk through this process of connecting the scientific question to the evidence and what's shown in the data. So I thought maybe I would just pause for a second and let you guys check it out and see if, and see if this sort of resonates with the way you talk about data in your classes already, or whether this is something that your students would find completely foreign and scary. So take a second at your tables and chat about what you think, and maybe we'll report back if we have a second still. Bring some to the back table there. All right, so I'm going to call on a couple volunteers maybe to talk about what is your experience with working with students and with data in the classroom. Is this something that you find the students wrestle with, or is it pretty straightforward?
I teach middle school science, so my kids have an 8th grade writing test, and I kind of paired up with one of the 8th grade writing teachers, and part of what they've got to do on the writing test is be able to support an argument. So we had them supporting arguments based off of the labs that they were doing. So it worked pretty well for us. But I had something very similar to this for them to do in the labs.
Cool. Awesome. Well, I hope maybe connecting with a real science case study might be. I'm sure you're probably already doing that as well. But
yeah, we were using the data that we were getting from our labs.
Okay.
But
Yeah. And that's something we do at the college level a lot too, is the students collect a ton of their own data in our teaching labs. We often don't do a good job of connecting them with the raw data from other studies. Yeah. Any other thoughts or impressions? Thank you for being brave and sharing. All right, well, I'm going to keep you going then on my journey through science briefly. You're very close to dinner. You're going to be even closer if I can't find my clicker. Okay, so this, I think, was one example of a really fantastic field-based research project that I got to do. When I came back to Virginia, I started a postdoc at VCU. Looking for any sort of project that connected with my theme of population biology, and I ended up sort of opportunistically connecting with a guy working on the invasion of the Gypsy moth. I'm a reptile amphibian person, I don't really know about insects, but I learned really quickly. It was a wonderful experience to get to learn something about a whole different group of organisms, and the Gypsy moth was a really fantastic invasion story. We know that the Gypsy moth was introduced in Medford, Massachusetts, and we know it was in 1870 or 1869, and we actually know the name of the guy who brought them over, and we know the address of his house, and he actually reported it to the game department that he had accidentally released his sort of caterpillars from silk production, and warned them that, hey, this could be a problem, and they spread and spread and spread and spread. And in case you think, oh yeah, gypsy moth, they've been here for a while. Well, these are all the forests left in the United States that are still susceptible to gypsy moth. And some people call the Ozarks the gypsy moth promised land. So we hope current efforts to keep them contained are successful. And so some of my work looks at, here in Richmond, we're at the southern edge of this invasion front, and we're interested in how hot can gypsy moths stand it to move further south. So then when I sort of reflect and look back at what I'm doing now, after I left VCU, I've been at University of Richmond for 3 years. I thought a lot about the experiences I had as an undergraduate, catching turtles, getting to present my results at meetings. And here I am, 10 years later, getting students to rear caterpillars in the lab and think about these questions, and it's been really amazing to provide these kinds of opportunities to students. And in my lab at the University of Richmond, we do a whole bunch of different projects, still gypsy moths and some other pest species of insects, a little bit back to newts, some baseline amphibian surveys up at 40p Hill. But then I want to tell you just a little bit about this redback salamander project, because potentially this might be a project that some of you or some colleagues that you know might be interested in joining. So many of you, who's seen a redback salamander? Anyone under law? A couple people? Oh, good. They are in your backyard probably. Maybe not if you live in a very urban environment, but they are very prevalent in Virginia. They're a terrestrial forest amphibian. They're located all up and down the East Coast, but they spend most of their time under rocks and logs on the forest floor. They're actually lungless, so they breathe entirely through their skin. So if it's not wet, they're not out. They can be active on the surface when it's raining, but who is out in the forest when it's raining except for people like me? Or they're underground in burrows most of the time. And around here, they're just reaching the end of their active season, and they go underground from May until about October. So you wouldn't see them on the surface now until it gets cool and wet again in the fall. So at University of Richmond, we're part of a big network trying to look and understand about how salamanders might respond to changing climates. So there's a whole network of people all up and down the East Coast and over into Michigan and Indiana and Ohio, who've set up study sites to try to understand how the populations of salamanders might change as we get different climates. And with the secondary goal of just having a better appreciation for the salamander species and its role in forest ecosystems. So our study site is actually located in the James River Park. I love getting my students out into an ecosystem right in their backyard. So we're right near Forest Hill Park. And we have these plots hidden away in the woods of coverboards. So it's really high-tech science here. It's just a little piece of I think it's like a two by something that we chopped up into pieces. And we have 50 of them, and we have 6 plots of these 50 boards in a 5-meter by 10-meter array. So they're actually really close together. And we see all sorts of things under these boards. We see insects, we see snakes, we see other kinds of amphibians. But our real, our creature of interest is this redback salamander here. So we go out and we mark them all, and we count them, and we look at when the population is active, and how these population sizes are changing. And the reason I mention this project in particular is that it's a big citizen science initiative as well. And so the network as a whole, and myself personally, are very interested in getting schools set up with some of these plots, because we think it's a great tool for seeing a lot of forest creatures and understanding and appreciating how these creatures might change in the future. So if you or a teacher at your school has a wooded area near you where you might be interested in setting up one of these plots, definitely let me know or get in touch. We'd love to bring you some boards. So it could be a cool project that we're trying to get going in Richmond area. So we've completed our first 1.5 years of sampling on this project. How much time do I have left? 5 minutes?
About 7 minutes.
7 minutes, okay. So I wanted to end by talking a little bit about what I do now at the University of Richmond in the classroom. So I teach an ecophysiology course on animals in extreme environments. So we talk about how animals survive deserts and cold environments. I teach the third course in our intro series on ecology, evolution, and physiology, and I teach field ecology. And I love any excuse to get our students into the campus lake. I catch turtles, which is really fun. We try to make sure no one loses a finger. And what motivates me to do this is, and it's kind of interesting being at in a sort of general biology department instead of in a wildlife department or an ecology department. We get a lot of pre-health students, so I sort of get students writing things on my evaluations like, well, I really hate ecology, but she was okay. So. No one likes learning about environmental topics, but it was a fine class, I guess. Sort of things like that. But when I teach things like field ecology, I get to identify the students who are really passionate about being outdoors. And I was just grading their field notebooks from this semester, and I hope she just put that in there for me. But it was really exciting to see students get excited about being outdoors, and that's what really ignites my passion in the classroom, is connecting students with real, authentic research and trying to get them outside to experience some of it themselves. I wanted to tell you some of my favorite classroom resources. For those days that you can't get outside, or those systems that you can explore, SimBio is a really nice resource. They have simulation-based labs and interactive chapters, mostly geared toward high school and undergraduate audiences. Also for more advanced audiences is Dryad Lab, where the Dryad is a repository for data. So you can, let's say you want to try to bring some data on Zika into your classroom. You can go to Dryad and actually find some of the latest scientific papers and the data from those papers and present it to your students. But they're not polished, right? It's not like data nuggets where everything's sort of worked out for you. So Dryad has started to make some educational modules from the data that's reposited in their database. Then some of the stuff I brought you guys on the table is from HHMI Biointeractive. I've gotten a little bit involved with trying to modify some of their resources for a college audience. Right now, they do a really great job of reaching out to high school classrooms, and they're trying to expand into college classrooms. Has anyone used HHMI resources? Okay, well, then I am probably preaching to the choir, but I really love their resources and the types of activities and videos. Everything is really well done. They have not only, they have a wide variety of topics, so it's not only environmental, ecology, and evolution, but it's also chemistry, genetics, cell, geology, so it touches on a wide variety of different scientific, bioinformatics, genomics, a lot of different topics their resources cover. And they have everything from films to interactive media to data points, classroom resources, animations. So here are some of their animations on DNA and replication, translation, actually getting to see these processes in action. And the nice thing about HHMI resources is that they're all free, as you know. A lot of their instructor resources have background information, suggested pause points in some of their films, resources for questions, student quizzes with answer keys. They have short films that range from 10 to 30 minutes that are all really well done. A lot of them draw on scientists and their research, multimedia, different click and learns, and interactives for students, as well as a few virtual labs. They only have, I think, about 8 right now, but I think they're expanding this repertoire as well. And some of my favorites are these Science at Work clips where you can connect with scientists and the projects that they're doing. And there's a partner site that they've partnered with called Science in the Classroom. I'll add that to the PowerPoint before I post it. They actually annotate scientific articles both for college and for high school classrooms, where instead of just the dense scientific paper, it breaks up the sections of the paper with background info and videos and details on the experimental design to really make it digestible for students. And also, my favorite, working with data, so bringing in statistics and math in digestible way for students and how to think about spreadsheets and data analysis. So here's some examples of their statistics information and how to do basic graphing, for example, in spreadsheet programs. And now with Google Sheets, a lot of this is accessible even for classrooms where you don't have access to Excel. And I'm always surprised, but I always get students in my class with Macs and they want to try to use numbers, and I don't know anything about numbers, or other different programs. So it's really good to be able to be flexible with the platform that the students are trying to analyze data in, and just some other working with data examples. I think that is the end. This is a newer one, is a citizen science project looking at camera tracking information. So they have camera traps throughout this national park, and your students can actually analyze data from these camera traps on the presence of big mammals in Africa. So students like me, who's their dream to go study animals in Africa, you can bring that into your class.